flight. That one's not scheduled yet. But all the black ones have already flown in space. The blue ones all have scheduled launches. This one is just a proposal at the stage. This one is finished but needs a launch. So a lot of interesting science gets into space this way. So I have a couple of slides to tell you about the GeneSat and PharmaSat experiments, kind of a one-slide overview. So GeneSat was our first experiment of this type. It grew E. coli in a dozen microwells on a microfluidic card, basically a laminated acrylic multi-layer fluidic card. The little white circles you see are nylon mesh filters that prevent E. coli from getting washed out of the wells because these are non-adherent cultures. So they have to be delivered about 6 weeks before launch. So they know we put them in a state of stasis by just putting them in a buffered saline solution. And once they get up in space, about 6 weeks later, then we replace that saline with a nutrient medium that contains sugar. E. coli take off and grow. And we look at green fluorescent proteins to look at expression of a constitutively expressed gene. And we look at optical density to look at cell population. So that was launched in December of 2006. The data came back a matter of days later, and these are the fluorescence versus time curves. Time goes, it's hard to see, but it goes out to about 50 hours here. This is what it looked like in space. This is what an identical experiment looked like on the ground. So you see they're very similar, but there are some differences. The blue curves versus red are two different strains of E. coli, so obviously the blue ones grow faster, and that's true, same on the ground and in space. If we look at the region of the growth curves for these, where they're growing exponentially, and put this on a semi-log plot and get a double